Second are cell junction, that is how the cells communicate with combined with one another. Then is cell communication, that is also known as cell signaling. So how do they transfer the signals to one another? Next are steps involved in the signal transaction construction. So how the signaling, what are the various steps that are involved in the cell signaling? And last is the forms of the intracellular signaling, as there are various types of cell signaling, intercellular as well as intracellular. So how a signaling takes place between the same cell and the between the different cells. So starting with the cell division, most almost all cells of our body undergo cell division because it is the process by which the cells reproduce themselves, or cell makes the copies of themselves. In simple terms, we can say that the cell division is the process by which a parent cell divides into two or more daughter cells. These daughter cells are similar to the parent cell. The parent cell is the cell which undergoes division, and it divides or it makes the exactly similar copies of its own. So if we take the daily life examples, like in human body, in a single day near about 2 trillion cells divide every day. Okay, so in the if we take the example of our skin, so in a single day we lose about nearly 30 to 40,000 skin cells every day. If we collect their mass, so it becomes that we shed over 0.5 gram of the dead cells in a single day. It's not that the cell divide due to the because the cells have to become larger. Okay, cells also divide so that the living things can grow. When an organism grow, it isn't because the cells are getting larger, but it's because the cells are dividing to produce more and more cells. So there are two types of cell division. The first type is known as somatic cell division, and the other is known as the reproductive cell division. So now, what is the difference between somatic and reproductive cell division? So before going into that part, we must know what is a somatic cell which causes the somatic cell division, and what is a reproductive cell which brings about the reproductive cell division. So the difference in these cells is, a somatic cell is that cell of our body which is other than a germ cell, or which is other than a reproductive cell. So, what is a germ cell? A germ cell is the cell which is also known as a reproductive cell, or also known as a gamete. It may be sperm in case of the males. So, male sex cells are also known as sperms, and female sex cells are known as oocytes. Okay, or it may be any precursor cell which is destined to become a gamete. Hence, we can conclude that a reproductive cell is known as a sex cell. In case of males, it is a sperm, and in case of females, it may be oocyte or ovum. Whereas a somatic cell is the cell which is other than a reproductive cell. So, the somatic cell division will take place by the somatic cell. So, in what happens in case of somatic cell division? In this case, the cell undergoes a nuclear division, that is known as mitosis, and it undergoes the cytoplasmic division, that is known as cytokinesis. By this process, the production of two similar cells takes place. Each is having the similar number and similar type of the chromosomes as the original cell. In case of somatic cell division, in daily life, it replaces the dead or injured cells and it adds the new cells or new ones during the growth of our tissues. Next is the reproductive cell division. So what is reproductive cell division? In this type, the gametes are produced. As we already discussed, gametes are the reproductive cells or also known as sex cells. These are required for the process of this division and these are needed to form the next generation of sexually reproducing organisms. So reproductive cell division, it unlike the mitosis, it consists of two steps. Wherever we have seen that in case of mitosis, it is a single step process. So this two-step division is known as meiosis, in which the number of chromosomes in the nucleus is reduced by the half. So, what are the functions of the cell divisions? Why do the cell need to divide? Okay. So, cell division is the crucial process for an organism to produce, to reproduce, or to grow and to repair. Hence, cell division has three main functions. The first function of cell division is the reproduction of an entire unicellular organism, as the organisms are of two types. First one are unicellular, uni meaning single, cellular meaning cells. Cellular organisms are organisms that have single cells and multicellular organisms are organisms that are having multiple cells. So in case of multicellular organisms, the function of cell division is to growth and repair of the tissues, and third function of this cell division is the formation of the gametes for sexual reproduction in the multicellular organism. Hence, we can conclude that the division does not take place only for the reproduction, it also takes place so as to growth and repair of the tissues. Now let's understand these divisions in detail. So firstly, let's focus on the somatic cell division. So the process of somatic cell division is an ongoing process. It starts as soon as the egg is fertilized. That means as soon as the meeting of the sperm and the ovum takes place and the zygote is formed, the cell division starts. What happens after the zygote formation? When it develops into the fetus in the mother's uterus, its cells will start multiplying. Okay. After multiplication, the each cell will adopt a special function, okay, which provide the sum of the body's total physiological functions. So the lifespan of every cell is very limited. Every cell becomes old and has to die after some time. So many cells which become worn out and which die, they need to be continuously replaced by the similar cells. And this process is known as mitosis. The organelles in the cytoplasm of the daughter cells are incomplete at during the beginning of the cell division. But after the cell is fully matured, the cell organelles are also matured. I hope you all are aware about the cytoplasm, which we have discussed in our earlier lecture. The fluid substance that is present in the cell, which consists of all cell organelle, is known as the cytoplasm.
So all the cell organelles are suspended in the cytoplasm of the cell. So when it is divided, when the new cell forms at the beginning phase, these cell organelles are incomplete. But after the cell is fully matured, the cell organelles will develop to the fullest. Next we have is the cell cycle. So what is the cell cycle? In cycle, there are the order of the sequences in which the somatic cell duplicates, or we can say the somatic cell replicates its content, and it divides this into the two. Okay, or we can say that in this the single parent cell forms the two daughter cells which are similar to one another and similar to that of the parent cell. So if we take example of the human cells, the human cell contains 23 pair of the chromosome. For they are in pairs. If we talk them singly, so there are a total of 46 chromosome in the human. So one member of each pair is inherited from each parent. So the chromosome which make up each pair are known as the homologous chromosome or homologs. Hence, we can say that the homologous chromosome, homo meaning same. Homologous chromosome are those chromosome which contains the similar or identical genes and arranged in the same order. Okay, homologous chromosomes are the similar chromosomes. The exception of this rule is one pair of chromosome called the sex chromosome. Hence, in our body, as we have discussed here, there are total 23 pair of the chromosomes, out of which 22 pair are similar, that are known as autosomes. But the last pair is known as sex chromosome. So these are designated as X and Y. X chromosome are slightly bigger and Y chromosome are slightly smaller in size. So because the somatic cell contains two set of the chromosome, they are called diploid cells, and these are written as 2N. Di meaning two. So there are diploid cells. So, when the cell reproduction starts, when the cell reproduces, it must replicate all its chromosome to pass its genes to the next generation of the cells. Hence, the replication or the duplication or the multiplication of the chromosomes is very crucial, as the chromosome contains the genes which contain the hereditary material of the cells, and this hereditary material is being passed from one generation to the next generation. So the cell cycle consists of two main phases. The first phase of the cell cycle includes the interphase, and the second phase of the cell cycle includes the mitotic phase, and it is also known as the M phase. The interphase is the one in which the cell is not divided, whereas the mitotic phase is when the cell is divided. So these are the phases of a cell cycle. So in a complete cell cycle, there are two phases, interphase and the mitotic phase. Interphase is further divided into the G1 phase, S phase, G2 phase, and the mitotic phase is divided into two types. That first one is mitosis and other is cytokinesis. In case of the mitosis, the nuclear division takes place, and in case of the cytokinesis, the replication of the cell organelles takes place and the cell is formed into the two. Next we have is interphase. So as I told you previously, in interphase there is no division. Okay. But there are a series of sequences like G1, S and phase G2. So during this phase, only the replication of the DNA takes place. So interphase is a state of high metabolic activity. During this interphase, the cell does its most of the metabolic activities like growing. So, what is the significance of these phases? G phases, G1 and G2. G, the term G stands for the gap. So, these are the gap phases in which there is no multiplication occurs. And the S phase is the one, S stands for the synthesis. In S phase, the synthesis of the DNA occurs. Thus, we can say the G phases are the periods when there is no activity related to DNA duplication as it occurs during the phase S, that is synthesis phase. Hence, the cells are thought as gaps or interruptions in the DNA duplication. So, G phase denotes the gap phase. This is the diagram of a cell cycle. So these are the different phases that are involved in a cell cycle. So here the purple color indicates the interphase and this phase shows the mitotic phase, that is M phase. So what happens in G1 phase? For example, we consider that the time for cell division is 24 hours. So during that hours, first 8 to 10 hours occur during G1 phase. So during this phase, the cell is metabolically active, it is highly activate, and the duplication of the cell organelles and the cytosolic component begins. Next is the centrosome replication. It also starts at the beginning of the G1 phase. As we already discussed in the structure of the cell, that the centrosomes are the structure which help in the division of the cell by producing the mitotic spindles. So this whole G1 phase is of 8 to 10 hours. Next phase is the S phase. So S phase lies between the G1 phase and the G2 phase. During this phase, the the synthesis, the replication of the DNA take place. It takes about 8 hours to do such. Next phase we have is the G2 phase. So G2 phase comes after the S phase and it is followed by the mitotic phase. G2 phase consists of 4 to 6 hours. During this phase, the cell growth continues and the enzymes and other proteins are synthesized. These enzymes and proteins are synthesized that are crucial for the activities of the cell. And the centrosome which started replicating during the G1 phase, their replication is completed at the G2 phase. Now the cell is fully ready for the division. That division phase is known as the mitotic phase or the M phase. This phase further consists of the four phases. They are named as prophase, metaphase, anaphase and the telophase. The process of mitosis we'll study in our next slides. After the mitotic phase, the last phase is cytokinesis. So in case of cytokinesis, the parent cell that was a single cell, it is completely divided into the two daughter cells having the same material as the parent cell. So this is the part where the replication of the DNA occurs, that is the S phase. So how the DNA replicates during the S phase or the synthesis phase. So how the two strands of DNA are synthesized from a single strand. Okay. We all know the DNA contains the helical structure, and in this the nucleotides are held together by the hydrogen bonding. So what happens here is, during this phase the hydrogen bonds are break broken. So during this phase, two strands of the double helix are separated by breaking the hydrogen bonds between the nucleotides. So here the hydrogen bonds are breaking, and the nucleotides, each nucleotide will uh, each nucleotide will attach its complementary nucleotide sequence. For example, T will be attached to the A sequence, or the G nucleotide will make the form with the C nucleotide. Hence the complementary nucleotides attach at the proper site, and a new strand of the DNA is synthesized alongside each of the original strands. Here we can say this was the old strand of the DNA, and here the complementary nucleotides, uh, the synthesis of these strands begins, and there is the whole new strand of the DNA. So from one DNA, now we have the replicated two new DNAs. So then comes the mitotic phase, or we can say the phase in which the division finally occurs. So mitotic phase, which is denoted as M phase, in this phase it results in the formation of two identical cells, means two similar daughter cells. It consists of two phases. First one is nuclear division, that is known as mitosis, and the other one is the cytoplasmic division, that is cytokinesis. So in nuclear division, the content of the nucleus, like the chromosomes, will start replicating, and in case of cytokinesis, the division of the cytoplasmic contents takes place. So in case of the nuclear division, that is mitosis. So in mitosis, it is the distribution of two set of the chromosome into the separate nucleus. This process results in exact partitioning of the genetic information. Hence, we can say that during the nuclear division, the division of the genetic material takes place. Mitosis is further divided into four different stages. The first stage is prophase, second is metaphase, third one is anaphase, and the last stage we have is telophase. So, what is the prophase? It is the first phase of the mitotic division. In case of the prophase, the chromosomes become denser. They condense into the X-shaped structures. We can see in the diagram. So the chromosomes condense and become the X-shaped structures, which can be seen easily under a microscope. So now each chromosome is composed of two sister chromatids. Sister means similar chromatid. So they containing identical genetic information. The chromatids which have a different genetic information, which are not similar, are known as non-sister chromatids. And now the chromosome pair up so that the copies of the chromosome 1 are together and copies of the second are together, and so on. Hence we can say that in the prophase, the pairing of the chromosome takes place. We can say in the diagram, the pairing of the chromosome takes place, and these chromosomes are held in the center by a structure, centromere. So at the end of the prophase, what happens is the membrane around the nuclear. Here in this diagram, we can see the nuclear membrane, fully developed nuclear membrane. But at the end of this phase, the membrane will start dissolving or start disappearing. After the disappearance of this membrane, the chromosomes will be released into the cell, and the mitotic spindle will also form. The mitotic spindle consists of the microtubules and other proteins. The mitotic spindle consists of a protein named as the tubulin. It extends across the cells between the centrioles. Here we can see the centrioles, two centrioles from which these proteins are extended, that are mainly tubulin proteins. And at the end of this phase, these centrioles will start moving opposite to one another. Now the next phase is the metaphase. So in metaphase, the chromosome line up at end to end along the center, or we can say equator of the cells. In previous diagram, here the centrioles were not exactly opposite to one another. But in this case, we can clearly see that the one centriole is present here and the another centriole is exactly opposite to the other. So the centrioles are now at opposite poles of the cell with the mitotic spindle fibers extending from them. So here we can see the mitotic spindles. So these mitotic spindle fibers attach to each of the sister chromatid. These are the sister chromatids. These are similar to one another and they are held together with the help of the mitotic spindles. So next we have is the anaphase. Anaphase is a third phase in which the sister chromatid are pulled apart by the mitotic spindle. In previously we have seen that these sister chromatid were held by the mitotic spindle, and in anaphase these are pulled apart by the mitotic spindles, which pull one chromatid to the one pole. Here you can see with the help of the diagram, these are the sister chromatid. So one sister chromatid is on the other side of the pole and the other sister chromatid is on the next side or the opposite side of the pole. So once they are separated from one another, the chromatids are termed as chromosome. So here after separation, these chromatids are known as the chromosome. Next, last mitotic phase is known as the telophase. So in telophase, at each pole of the cell, a full set of chromosomes gather together. So here the full set of chromosomes will start gathering together, and the membrane which was dissolved during the first phase of the mitosis, that was during the prophase, it will start developing again. So the nuclear membrane reforms, that means it will be developing again. So a membrane forms around each set of the chromosome to create why? To create the new nuclei, and the mitotic spindles breaks up. Here we can say there are no mitotic spindles. At last is the cytoplasmic division. So in the cytoplasmic division, that is known as cytokinesis. In this phase, the division of the cell's cytoplasm occurs and organelles are also separated into two identical cells, that is known as cytokinesis. So when the cytokinesis is completed, then the interphase begins and the cell cycle will continuously repeated. Hence, we can say the sequence of events can be summarized as: firstly, the G1 phase occurs, it is followed by the S phase, then G2 phase occurs, then mitotic phase, and at last is the cytokinesis. Now let's revise it. What happens in G1 phase? All the cell is ready for the metabolic activities. The centrosomes will start replicating. and during the S phase the synthesis of the DNA takes place, or we can say the replication of the DNA takes place. During the G2 phase the completion of the centrosome occurs, and during the mitosis phase the complete division of the cell takes place. And last step to mitosis is the cytokinesis. In the cytokinesis, as we can see in the telophase, when the nuclear membrane again fully reforms, then we can see each cell has the same genetic material as was in the parent cell. Now these cells are pinched off from one another. This cleavage is known as the cleavage furrow. So the cells are pulled apart from one another, and then we can say from a single parent two resulting daughter cells are formed. So here is the complete process of the mitosis, that is step by step. First step is the interphase. So during this phase only the DNA is copied. During the second phase, that is known as the prophase, in this phase the chromosomes are paired up. Here we can say these are the single chromosome. Here the pairing of the chromosome into the structure like X takes place. Here the centrosomes were not at the opposite poles to one another, but during the prophase the centrosome will start moving at the opposite poles. Then it is followed by the metaphase. Here the mitotic spindles are also present, and the chromosome line up at the equator, so chromosome will place at the center of the cell. And during the anaphase these are pulled apart at the opposite poles, so these sister chromatids are pulled apart. And last is telophase and cytokinesis. So during this phase the cell pinches in the middle, which results in the two identical daughter cells. So that was all about the somatic cell division. Now take a look at the reproductive cell division. So what is reproductive cell division? This is the process of cell division which is carried out by the reproductive cells, and these cells are known as the gametes or the sex cells. So these gametes are the sperms which are matured in the testes of the male, and the ovum which are matured in the ovaries of the female. So unlike the mitosis, in the meiosis, four daughter cells are formed after the two divisions. What we have learned now that in case of mitosis only two daughter cells were formed from a single parent cell, but in case of the meiosis there is the formation of four daughter cells. Why? Because there are two divisions in the meiosis, that is meiosis 1 and meiosis 2. Hence, all the daughter cells produced are different from the parent cells. What we have seen in case of mitosis, that the parent cell and daughter cell all share the same genetic structure or the same cytoplasmic contents. But in case of the meiosis, all the daughter cells are different from each other, as well as they are different from their parent cell from which they are formed, and this is the basis for the uniqueness in the organisms. So during meiosis the pair of the chromosomes separate from one another, and each pair move to the opposite pole of the parent cell. So hence, when the cell divides, each of the daughter cell has only 23 chromosomes. So 23 number is the haploid number. Okay. This means when the ovum is fertilized, so it has 23 pairs, and the sperm also has 23 pairs. When the fertilization takes place, that means the meeting of the sperm and the ovum takes place, the resultant zygote that is formed, it has a full set of the 46 chromosomes. So these 46 chromosomes comes from the 23 comes from the male side, and the next 23 comes from the female side, which then make the 46 chromosomes, and this 46 is now the diploid number and 23 was a haploid number. Hence, half from the father and half from the mother. Next is when after the fertilization child is born. So a child has some characteristics inherited from the mother, and some characteristics will the child will inherit from its father. They may be color of hair, color of eyes, height, facial feature, and many diseases. Okay, so all of this depends upon the number of these chromosomes. Determination of the sex depends upon the particular pair of the chromosomes that are known as the sex chromosomes, that are X and Y. So, in case of the female, both the sex chromosomes are the same size and shape. These are known as X chromosomes. But in case of male, there is X chromosome that is larger and a smaller chromosome that is known as Y chromosome. When the ovum is fertilized by an X bearing spermatozoon, so if the X chromosomes comes from the father side, then the child born is female, and if the fertilization occurs by the Y chromosome that comes from the father side, the child is a male. So here we can see the if sperm contains X chromosome and ovum has X chromosome. In case of ovum, it has XX chromosome. The chromosome will always be X, but in case of the father, either it may be X or it may be Y. If the X and X will fertilize, then the child will be XX. That means the child will be female. So if the sperm has Y chromosome and the ovum has X chromosome, then the child will be XY. That means the child will be a male. Next we have the process of meiosis. So unlike mitosis, which is completed after a single round, meiosis occurs in the two stages that are named meiosis 1 and meiosis 2. So, during the interphase that precedes meiosis 1, the chromosomes of the diploid cell start to replicate. So, we can say in meiosis 1, firstly, the chromosomes will start multiplying, replicating. As a result of this replication, each chromosome consists of the two sister chromatids. What are sister chromatids, which we studied in mitosis? These are the genetically identical set of the chromosomes. Hence, they are attached in the middle with the centromeres, the structure known as the centromeres. They are X-shaped. This replication of the chromosome is similar to one that proceeds in the mitosis in the somatic cell division. So, in meiosis 1, it also consists of four phases similar to the mitosis 1. The first phase is known as prophase 1, second is known as metaphase 1, third is anaphase 1, and last one is the telophase 1. So, what happens in prophase 1? In prophase 1, the chromosome are shortened and thickened, or we can say they become condensed, and the nuclear envelope and nuclei will start disappearing like in mitosis. And further, the formation of the mitotic spindle occurs. But It is different from the mitotic in two aspects. That is, first one is the two sister chromatid of each pair of the similar homologous chromosome pair off. This event is known as synapsis. So in synapsis, the pairing of the similar chromosomes takes place. This results in the four chromatids from and with they form a structure known as tetrad. Here you can see in the diagram, these are the homologous chromosomes. So when their pairing takes place, that is known as synapsis. So this resultant structure is known as a tetrad. Tetra meaning four. So there are now four chromatids, that is tetrad. Secondly, it is different from mitosis in the aspect that the in the second part, parts of the chromatids of two homologous chromosomes may be exchanged by one another. Here, here these chromatids exchange the genetic material with one another, but these were not similar with one another, so these are known as the non-sister chromatids. Okay, so this pairing of the non-sister chromatids with the sister chromatids, that is termed as the crossing over. So it is known as the crossing over. We can say that these are crossing on the surface of one another. Okay, so this is the beginning of the prophase 1. In this, firstly the centrioles will start moving on the equators, on the opposite end of the equators. Similarly, as the mitosis, the mitotic spindle will form, and the sister chromatids are separated at the center with the help of the centromere, held together with the help of the structure that is centromeres. Then we can say that here the nuclear membrane was fully developed, but here it will start dissolving, and the synapses takes place. That means the uh, duplication of this chromatids takes place, which results in the formation of a structure that is the tetrad. So here these were the sister chromatids, but here the pairing of the non-sister chromatids, like the blue one is paired with the red one. So here the pairing will start, which will cause the crossing over between the non-sister chromatids. So this crossing over is the region. That's why all the daughter cells in case of the meiosis are different from the one another, because their genes are exchanged. They were not similar. Next phase we have is the metaphase 1. So in case of the metaphase 1, the tetrads which were formed by the previously phase, so they line up along the metaphase plate of the cell, or we can say that they line up at the equator of the cell, as you can see in the diagram. So with the homologous chromosomes side by side. So they are at the equator and the homologous chromosomes are side by side. Next phase we have is the anaphase 1. So during the anaphase 1, the membrane of each homologous pair of the chromosome separates, and they are pulled to the opposite poles of the cell by the microtubules which are attached to these centromeres. Here centromeres are seen, which are of green colors. So they pull these chromosome towards their selves. Hence, the separation of these takes place, and the paired chromatids, which were held by the centromere, they remain together. But in case of the mitotic phase, the sister chromatids were separated. But here, these chromatids are together. Hence, the last phase is telophase and the cytokinesis phase. So these are almost similar to the telophase of and cytokinosis of the mitosis. That means here the nuclear envelope will reform, and the content, cytoplasmic content will exactly divide. So we can conclude that the net effect of the meiosis 1 is that each resulting cell contains the haploid number of the chromosomes. Why? Because it contains only one member of each pair of the homologous chromosomes present in the starting cells. Hence, they are the haploid in number. Next is the meiosis 2. So it is the second stage of the meiosis, which is followed by the first stage, that is meiosis 1. So what happened in this case? It consists of the four phases as like the meiosis 1. So these phases are prophase 2, metaphase 2, anaphase 2, and it is followed by the fourth phase, that is telophase 2. So all these phases are similar to the phases that occur during the mitosis that we studied in detail in our previous slide. Similarly, the centromeres will split and sister chromatid will separate and they will be moved toward the opposite poles of the cells, like in case of the mitosis. So we can conclude that in meiosis 1, it begins with the diploid starting cells and ends with the two cells. Okay, here we have seen that in meiosis 1, the resulting cells were 2. Each cell was having the haploid number of the chromosome. But during meiosis 2, which is the another second round of the meiosis, in this case, each of the haploid cells are formed and here again the cells will start dividing. So the net result is the four haploid gametes, and all of these gametes are genetically different from one another and they are different from the original diploid starting cell. So this is the whole process of the meiosis. Okay. So it consists of two phases as I explained. This is phase 1 of the meiosis and this is the phase 2 of the meiosis. So firstly, in prophase 1, what happens is the nuclear membrane will start dissolving. Its envelope will start getting uh, detached from its membrane. Hence, the centrioles here they are shown, they will start producing the mitotic spindles. After this, these centrioles will move to the opposite ends of the poles, and here these chromatids are multiplied. This is known as synapses. After the firstly the synapses of the sister chromatids takes place. After that, these are crossed over with one another, that are they are crossed over with the known sister chromatids, like here you can see that the blue one, this is these are the sister chromatids. So this is one chromatid, this is another chromatid, this is, so these two are the sister chromatid, and this if we compare the red one with the blue one, these are the non-sister chromatids. When they cross over with one another, what we can say, the genetic recombination will takes place. That means now they have the variation in the characteristics from the one another, which is the main reason why in meiosis the daughter cells are completely different from their parent cell. So here during prophase 1, the tetrad which we can see here is one chromatid, second, third and fourth. So the tetrad is formed and these are at the center, these are held by the centromere. So after the crossing over takes place, then comes the metaphase. So in this phase, these chromatids come at the center of the cell, and after that their division will takes place. So these will be pulled apart with the help of these centrioles, with the help of the mitotic spindles. So this pulling apart will cause the separation of the homologous chromosomes. Homologous means same, chromosomes. So they will be separated. So it will be completely separated in the telophase, and what we see in the telophase, the nuclear membrane will again reform. So after that, it is followed by the meiosis 2. That means the second phase of the meiosis. So in second phase of meiosis, it is further the same cycle will repeat. Firstly, these chromatids will align at the center. Then further they will be pulled apart, and further from one cell, two daughter cell will formed. Similarly, two cells will formed here and two cells formed will be there. So the resulting cell will be four daughter cells, which are completely uh, different from their parent cell. So next we have is the cell cycle regulation. Okay, I hope now you all know what is a cell cycle, what does it contain. So this is the cell cycle which consists of the interphase, that is G1 S and G2 phase. It is followed by the mitotic phase, that is the M phase. So cell cycle regulation in this, these are regulated by the cell cycle checkpoints. So what are the checkpoints? There are different checkpoints in a cell cycle which make sure that the cell cycle is going on smoothly, that they check there are whether there are any nutritional requirement for the cell, or whether the replication of the DNA is okay. And so that's why these are known as the checkpoints. So they check out the process of the cell cycle completely. Hence we can define the checkpoint as it is the stage in which the eukaryotic cell examine the internal and external cues, that is the cell examining the internal and external signals. It decides whether or not to move forward with the division. So we can say that in case the checkpoints are put into the cell cycle to check whether the cell is okay, whether the all the requirement that are needed for cell division are okay or not. Okay, so there are a number of checkpoints which includes the three main important one. First one are G1 checkpoint, second are G2 checkpoint, and third one are the spindle checkpoints. G1 checkpoints are occur at the stage of G1S transition. The meaning of G1S transition is the cell is going from G1 to the S phase. Transition is, that means it is marching towards the S phase of the DNA synthesis. So here we can say the G1 checkpoint. So it occurs at the last of the G1 phase. And there is one more thing. There is the G0 phase, that is known as the resting phase. G0 phase is for the non-dividing cells. If the cells for any reason they are not dividing, so they go into the phase that is known as G0 phase. The example of this G0 phase is the neurons. In most of the cases, neurons cannot they do not divide. They, once they are produced, they are unable to divide, hence they go into this phase that is known as G0 or the resting state. Another checkpoint we have is the G2 checkpoint, which occur at the end of the G2 phase and before the beginning of the mitotic phase. Last we have spindle checkpoint. It occurs at the transition from metaphase to the anaphase. So what are metaphase, anaphase? It occurs at the mitotic phase. These are the last middle and second last phases of the mitosis. So the G1 checkpoints checks whether the nutritional requirement, the growth factor requirements, or they check for any DNA damage in the cell. If there is any defect in that, the cell will not be dividing. Similarly, the G2 checkpoint checks for the size of the cell, the DNA replication, that whether the DNA is replicated properly or not. Similarly, there is the metaphase checkpoint which checks for the chromosomes and the spindle attachment. That whether the spindle formation is okay, that whether the contents are pulled apart equally, and all that. So, what happens in G1 checkpoint? G1 checkpoint is located at the end of the cell cycle's G1 phase, just before when the cells start entering into the S phase. So, like I told you earlier, so they make the decision whether the cell should divide, or whether it has to delay its division, or whether it has to enter into a resting state, that is the G naught state. So, the condition which it checks are the nutrients and enzyme requirements for the DNA synthesis, growth factor requirements, hormonal requirements. Okay. So, these may be local conditions also, that are amino acid, glucose, oxygen, etc. So, if a cell that does not meet all the requirements that are mentioned above, then the cell will decide not to progress into the further phase, that is S phase. So at that point the cell division will be stopped. So whenever the cell will decide not to divide, the cell will go into the G naught phase, that is the resting phase, and it will further wait for the signals when the conditions are improved. If the conditions will improve, the cell can again start the division cycle. Next checkpoint we have is the G2 checkpoint. The G2 checkpoint is the also known as the G2 or G2 mDNA damage checkpoint. Why? Because it occurs after at the end of the G2 phase and before the mitotic phase. So its main function is to check whether there is any DNA damage or not, whether the replication of the DNA is okay. So if there is any impairment in the DNA replication, then the cell will not divide. If anyhow the defective DNA replicates, or the cell division continues, it will not, the cell will not last long, and it will lead to the apoptosis or the cell death immediately after the division, because the faulty DNA was there. So it causes the cell to die. Next we do have is the spindle checkpoint. So what is a spindle checkpoint? This checkpoint is also known as the metaphase to the anaphase transition. Okay, what is metaphase to anaphase transition? In the mitotic phase of the division, in the M phase of the division, it when the cells start from moving from metaphase, in which the cell the chromatids will align in the center, to the anaphase, in which these are pulled apart one from one another. So it is known as the spindle assembly checkpoint, or SAC, or the mitotic checkpoint. So it is a cell cycle checkpoint during mitosis or meiosis, which prevents the separation of the duplicated chromosomes. Why? Because the separation of these chromosomes occur at the anaphase, that is the second last phase of the mitosis. So until each chromosome is properly attached to the spindle, so we can say it makes sure that the chromosome are completely or properly attached to the mitotic spindle, and their division is equally done. Next we have is cell death. Okay. In earlier slides, we have studied that how a cell divides. But in that case, we will study that how a cell leads to the cell death. What happens to the cell that it has to go through the process of the cell death? So we can define cell death is the event of the biological cell ceasing to carry out its function. Ceasing means stop. Whenever the cell will stop carrying out its biological functions, then the process of cell death occurs because the cell is no longer functionally active. So it may result from the natural process, like whenever the cell gets old and dying, or whenever it need to be replaced by the new cells, or whenever it may result from such factors like disease, whenever a disease is there and whenever a local injury is there. So the death of the organism which the cells are part. So cell death is of following types. It may be type 1 cell death, that is known as apoptosis. Apoptosis, and part 2 is known as the autophagy. Both autophagy and the apoptosis are known as the programmed cell deaths, in which our body or designs the programmed cell, that body decide that this cell is defective, or it there is a spread of any infection or any injury, that this cell needs to die. Another one we have is the necrosis. So what happen in necrosis? Necrosis is the unprogrammed cell death. So in necrosis there may be the sudden death of the cells. So, the necrosis results from any injury, or it may result from the any process of infection. It is a non-physiological process. So, with that, our cell division is completed. I hope you all understand about the cell division. So, what are the processes in the cell division? So next we have the cell junction. So what do you mean by a cell junction? What are cell junction? So a junction, what is a junction? A junction is a space or between anything, or a junction is a space where the cells are meet. Okay. So junction are the point where the two things are combined or they communicate with one another. Cell junction may be it is a cell to cell communication, or it may be cell to the extracellular connection within the tissue or a multicellular organism. Or we may define cell junction are the contact points where between the plasma membrane of the tissue cells. So what do you understand in cell junctions? Whether a cell can connect with another cell, a plasma membrane of one cell can connect with another cell, or the cell can connect with the extracellular matrix. So cell junction is of 5 main types. First one are tight junctions, second are adherent junctions, third are desmosomes, and fourth are hemidesmosomes, and the last are gap junctions. Okay. So first one we have are the tight junctions. As the name suggests, tight junction, what will be the tight junction? So in tight junction, two cells will combine very tightly with one another. So there will be no gap. So like these are the cells and these are the cells, if they have to combine, so they will fully combine with one another. There will be no gap, as you can see in my fingers, there are no gap to pass anything across them. Okay, so what does it look like? Tight junction consists of the web-like strands of the transmembrane proteins. During the cell junction, remember one thing: all the mainly the cell junction are the exclusions of the plasma membrane. So the plasma membrane contains various protein molecules that are extended from the plasma membrane. So these extension combined with the another cells or the plasma membrane of the another cells, and they make the cell junction. So They consist of the strands of the transmembrane proteins, that are the proteins which are present in the surface of the plasma membrane are known as the transmembrane proteins. They fuse together to the outer surfaces of the adjacent plasma membrane. So they combined together to the cell surfaces to the adjacent, that is neighboring plasma membrane, to seal off the passageways between the adjacent cells. The examples are the cells of the epithelial tissue which lines the stomach, intestine and urinary bladder, and they have many tight junctions. Now why the tight junction are present in these sites mainly? Because through the stomach, intestine, bladder, they are the structures where the uh, things we eat or the food has to pass through, or we can say in case of urinary bladder where the urine has to pass through. So there are a lot of chances that they may be leak towards the cells. So to prevent the leaking or to inhibit the passage of these kind of substances, the tight junctions are present in these structures. Okay, so what is their function now? They prevent the contents of the organs which are mentioned above from the leaking into the blood or leaking into the surrounding tissues. This is the structure of a tight junction. Here you can see there is no gap visible. Okay, so these are the plasma membrane of the adjacent cells. These are the strands of the transmembrane proteins. So they held each cell in close contact with one another. Next, we have our second type is adherent junctions. So, what are adherent junctions? Adherent junctions contains the plaques. What is a plaque? A plaque is a dense layer of the proteins. It is nothing but a layer of protein on inside of the plasma membrane. The function of plaque is to attach the both the membrane protein and the microfilament to the microfilament of the cytoskeleton. So the plaque helps to attach the proteins that come from the membrane, plasma membrane, to the microfilament that comes from the cytoskeleton of the cell. So transmembrane glycoproteins here are known as the cadherins, which joins the cells. Each cadherin inserts into the plaque from the opposite side of the plasma membrane. So, suppose this is one cell and this is another cell. What we have studied in the center, there are proteins, layer of proteins known as plaque. So, these are two cells in center, plaques are present, and from these plaques attach the cytoskeleton and microfilament to these membrane proteins, and there are other transmembrane glycoproteins present that are known as cadherins. So between these two membranes in the center, cadherins are present, and these cadherin molecules help to join the cells with one another. So this is the structure of a adherent junction. This is the plasma membrane of the one cell. This is the plasma membrane of the another adjacent cell. So here we can see that in the center there is the formation of the plaque. Plaque is nothing but the protein. And in the center the transmembrane glycoproteins are present. So here we can say this is the transmembrane protein of the neighboring cell, this is the transmembrane protein of the another cell. So here these protein molecules that are known as cadherin, they combine with one another and they held the cells together. This is the intercellular space, that is the space between these two cells. So each cadherin insert into the plaque from the opposite side of the plasma membrane, as we have just seen in the diagram. So it partially crosses the intercellular space and connects to the cadherin of the adjacent cells. Yes, it will partially connect the cadherin of the another cell. Here is also a structure that is known as the adhesion belt. So this is the adhesion belt which surrounds the cell in the middle. So this adhesion belt also contains the microfilament that are known as actin. These are the contractile proteins which are present on the adhesion belts. So, in case of the epithelial cells, these adherent junctions form the extensive zones called adhesion belts because they encircle the whole cell in a similar fashion. We tie a belt around our waist. Okay, like we tie belt around our waist. Similarly, in case of the cells, adhesion belts are present which completely encircle a cell. So these adherent junction helps the epithelial surfaces to resist the separation during the various contractile activities. Like when the contraction takes place, when the contraction happens, the movement takes place. So these belts, these adherent junctions held together the cells that it resist the separation between the two. Example is when the food moves through the intestine. Okay, so from this we can say that these are present in the intestine. Whenever the food moves through the intestine, the process of the movement takes place. So these adherent junctions make sure that the cells do not separate from one another. So the next junction, the third type of junction we have there are the desmosomes. Desmosomes resemble in certain ways with the adherent junctions, like they also contain plaque. The term desmos comes from the desmo, that desmo means a band. So they contain a plaque and they have a transmembrane glycoprotein which are similar to the adherent junction. Here also the glycoproteins are same, that are cadherins. So these cadherin extend into the intercellular space between the neighboring cells or adjacent cell membranes, and they attach the cells to the one another. However, they are different from adherent junction in the aspect that the plaque of the desmosomes does not attach to the microfilament. What we have seen in the previously adherent junctions, that the plaque contains the microfilament, that were the contractile proteins that were composed from the protein actin. But in case of the desmosomes, the plaque is attached to the cytoskeleton known as the intermediate filaments. These intermediate filaments are composed of a protein. That protein is known as keratin. So this is the structure of the desmosome, in which the two plasma membrane, the two adjacent plasma membrane or two neighboring plasma membranes are held together by the protein, the transmembrane protein that are known as cadherin. Okay. Here is the plaque, presence of the plaque, and in this plaque, instead of the microtubules like in adherent junction, here we have another filament that is known as keratin. It is the intermediate filament. So what is the function of these intermediate filaments now? These filaments extend from the desmosomes on the one side of the cell and across the cytosol of the desmosome to the opposite side of the cells. Hence, what happens in this case? The desmosome, the intermediate filament from one cell goes to the opposite cytosol, from cytosol of one cell goes to the opposite side of the another cell. So this structural arrangement contributes the stability of the cells and tissue. So it makes the cells and tissue more stable. These junction desmosomes, they are very common among the cells which make up the epidermis and among the cardiac muscles cells in the heart. So what is epidermis? Epidermis is the outer layer of our skin. Our skin is divided into two layers, that are epidermis, that is outer layer, and the dermis, that is the inner layer. Okay, so they form the epidermis of the skin, and another structure which they form are cardiac muscles in the heart, that are that function like the contractile muscles. So another function of these desmosome is that they prevent the epidermal cells from separating under the tension. Whenever there is a condition of any tension onto the surface of our skin, epidermal cells, so they prevent these tension. They resist this tension, not only to the epidermis, also to the cardiac muscle cell from pulling apart during the contraction. Next structure we have is hemidesmosomes. The word hemi means half, desmosome means desmosome. Hence, we can say that hemidesmosomes resembles the desmosomes. So they are quite similar to the desmosomes, but they do not link to the adjacent cells. Um, the junction which we have studied earlier, all we have seen that they were linking one cell to the another cell, but here is one exception. In this case, in case of hemidesmosomes, which are half resemble to the desmosomes, they do not link the neighboring cells. Instead, they link the plasma membrane to the basement membrane of the tissues. What is the basement membrane? In my next session, we will be studying in detail about the tissues. So what are tissues? These are the group of the cells. So the group of the cells that are tissues, so they are held together by a basement membrane. Firstly, the basement membrane is present. On that, the various different cells are present. Number of cells are present that are termed as tissues. So the name of hemidesmosome arises from the fact that they look like a desmosome. However, their transmembrane glycoproteins are different than that of the desmosome. Here, the transmembrane glycoprotein, that means the glycoproteins which are present in their plasma membrane, they are integrins rather than caderins. Okay. In case of desmosomes, what we had seen that the transmembrane proteins were caderins. Here, there is another protein, transmembrane protein, that is known as the integrin. So, on inside of the plasma membrane, these integrins attach to the intermediate filament, like in desmosomes. Here, also the intermediate filaments are composed of a protein named keratin. So, this was the internal inside of the plasma membrane. But outside the plasma membrane, what happens on outside? The integrin attached to the protein called as laminin. Laminin is the protein which is present in the basement membrane. I explained you what is the basement membrane. So here the protein, that is laminin, it is attached with the integrin, that are transmembrane proteins of the plasma membrane, and it is attached to the laminin of the basement membrane. Hence, we can say that in case of the hemidesmosome, the plasma membrane are of one cell are attached to the basement membrane of the tissues. Thus, hemidesmosomes anchor cells not to each other. That means they only not join the cells with one another, but rather they also join the basement membrane to the plasma membrane, as I explained. So this is the structure of the hemidesmosome. So here we can see the plasma membrane. These are the protein that are known as integrin. These are different from the previous protein that were the caderin. Okay, these proteins are attached with the laminin protein of the basement membrane. Here also is the formation of the plaque in the center and the extension of the intermediate filament that are keratin from this plaque. Next, we have our gap junctions. So, like the name indicates, what are gap junctions? These are the junction, or these are the communication points where there are the gaps. Unlike the tight junction, these are completely opposite with one another. In tight junction, what we have seen, there was no gap at all. But in case of the gap junction, there is the gap. So from this gap, very molecules or very small ions can easily diffuse through. So a gap junction is a membrane protein called connexin, and it forms a tiny fluid filled tunnel that are known as connexons. Okay. The function of connexon is to connect the neighboring cells with one another. The plasma membrane of the gap junction are not fused together as in the tight junction. This thing I explained you already. So, but rather than tightly held with one another, they are separated by a very narrow intercellular gap. So, there are intercellular gaps present among the cells. Through the connexons, these are the fluid filled tunnels. These connections are the channels, they serve as channels through which the ions or various small molecules can easily diffuse from the cytosol of the one cell to the cytosol of the another cell. But the passage of the large molecules such as the vital intracellular protein is prevented. Hence, we can say that from the gap junction, only small molecule or small ions can diffuse easily, but not the larger molecules. Next, the function of gap junction is to transfer the nutrients and perhaps the cellular waste. So they transfer the nutrients as well as the cellular waste, especially in case of the avascular tissues. What are avascular tissues? Which do not have the vessels. Such tissues are lens of the cornea of the eye. This is the example of a gap junction. Here we can see the adjacent plasma membrane have some gap between them, and here the proteins are known as connexons. These connexons are composed of the connexins. Here we can see the gap between the cells. That's why they are named as the gap junctions. Now the function of the gap junctions, they allow the cells in a tissue to communicate with one another. Example is in a developing embryo. When the embryo is developing, some of the chemical and electrical signals that regulate growth and cell differentiation travel via the gap junctions. So they also help in the movement of the signals. That signal may be chemical or electrical signal. Next function is these junctions also enable the nerve cells or muscle impulses to spread very fastly between the cells. So this is the process. This nerve impulse or muscle impulse is the process which is very necessary for the normal functioning or operation of some parts of the nervous system, and for the contraction of muscles in the heart, the gastrointestinal tract, and as well as the uterus. This is the whole diagram. In this diagram, all the junction are shown. Like first one are tight junction. Okay, so they are present here between the adjacent cells. Here they do not have any gap and they are tightly packed with one another. Here you can see the adherent junction. The blue color is shown. These are the adhesion belts. So adhesion belts are clearly seen on the surface. Okay. So next we have the desmosomes. So desmosomes, in the desmosome, the transmembrane are cadherin molecules, and there the intermediate filaments keratin are present. So what is the difference between all of these junction? You have seen their location, but what is the difference between them? Okay. So in first junction, you can see there was no gap. They were cells were very tightly packed with one another. But in case of the second junction, that are termed as the adherent junction, in these junction, a belt is present, that is adhesion belt present. On the surface of these adhesion belts, the microfilament that are composed of the actin, these are present. And here the transmembrane proteins are the glycoproteins that are known as cadherin. These cadherin molecules are similar in case of the third type of the junction, that are known as the desmosomes. Okay. So the plaque is present in three types of the junction, in the second type, third type, as well as in the fourth type, but they only differ in the transmembrane proteins. Like in case of hemidesmosome, the transmembrane proteins are entirely different. These are the integrins, and there the cells are attached with the basement membrane rather than with the another cell. So last one we have gap junction. So this is the junction in which the uh, cells are having some gaps in between them. Next we have is cell communication. So what do you mean by cell communication? Cell communication is the process with which the cells receive signals, or cells send signal to the one another. So all organisms in our body need to respond to their changing environment in order to survive. If a cell has to survive, it need to send signal to the another cell. So these responses are governed by the ability of the particular cell to sense the physical changes, or as well as to sense the chemical changes or chemical signals, chemical cues surrounding them. So the process of this sensing and responding to the extrinsic signal is known as the cell communication. The cell communication is also known as the signal transduction or the signaling. We can simply call it as the cellular signaling. So, a cell responds to a variety of extrinsic signals that include the chemical messengers, it may be hormones, the growth factors, the neurotransmitters, etc., or it may be electrical impulses, mechanical forces, pH, heat, or the light. So next is the type of the cellular signaling. So we have two main types of the cell signaling, that is intercellular signaling, and the next is the intracellular signaling. The term inter means between the cells. The term intra means within the cells. Hence, we can say the intercellular signaling, it includes the communication between the cells, and the intracellular signaling involves the signaling changes that occurs within the cells. So next we have the steps involved in a signal transduction. So what are the steps that involved in the cell signaling? So firstly, before that, you need to understand about the sending cell and about the target cell. So what is a sending cell, what is a ligand, and what is a target cell? A sending cell is the one who sends the signals to the another cell. The cell which receives this signal is known as the receiving cell, and it may also termed as the target cell. And this target cell is having the receptors present on its surface, because the sending cell releases the chemicals, these are termed as the ligands. So these ligands bind to the receptor present on the target cells. Okay. After these signals bind to the receptor, then they will cause the initiation of the signaling. But the cell which do not have the receptor for this particular ligand is known as a non-target cell. You need to understand that not every cell has the receptor for the particular ligand. Okay, so the cell which do not contain the receptor for a specific ligand will be known as the non-targeting cell, or we can say it the non-receiving cell. Hence, the first step is a signaling molecule known as the ligand or a primary messenger. It is produced by a signaling cell. So signaling molecule or a ligand or a primary messenger, that it is produced by a sending cell. So these ligand bind to the receptor which are present on the surface of a target cell. Then next step, the binding of this ligand to the receptor brings about the conformational changes in the receptor, and it activates the receptor in turn, which initiates the signals. So in the third step, on the activation of the receptor, the response is visible in the target cell. After the generation of the response, it includes changes in the cellular metabolism, and the gene transcription, synthesis of the proteins can take place, and so on. Next, we have the forms of the intracellular signaling. The intracellular signaling means the signaling between the cell. So it is divided into the four main types. First type is autocrine signaling. Second we have is paracrine signaling. Third we have is endocrine signaling, and at last we have is synaptic signaling. Now let's study these signaling one by one. First we have is autocrine signaling. So the term auto means self. So the meaning of autocrine signaling is one in which a cell targets itself, or we can say in autocrine signaling a cell may send signal to itself, and by doing so it releases a ligand that bind to the receptor on the surface of its own. You can see in the diagram. So this is the sending cell. Here the sending cell and the receiving cell, they both are the common. Here this cell is sending the signals, and again it is also receiving the signal. In other words, a signaling pathway in which the secreted substances exerts its activity on the same cell from which it was produced is known as the autocrine signaling. The example of the autocrine signaling includes the tumor cells or the cultured cell which are cultured in the lab. Next we have is paracrine signaling. So paracrine signaling is also the signaling which targets the nearby cells, or which targets the neighboring cells. So in this signaling, it is also communicates over a short distance. So signaling in which the substances are produced by signaling cell, it diffuse to nearby target cells for generating a response is termed as the paracrine signaling. In this type of signaling, in which the cells communicate over the very short distances. What are the functions of the paracrine signaling? It allows the cells to locally communicate or to locally coordinate the activities with the neighboring cells or with the adjacent cells. The paracrine signals are especially important during the development, when they allow one group of the cell to tell a neighboring group of the cells what cellular identity is to take on. Now what is the meaning of this line? While the development of a child or a fetus is going on, all the cells do not has a specialized function. At the starting stage, the cells do not are not specialized. They are unspecialized cells. That means they do not have a specific function. So during that time, the paracrine signaling is very important because it tells its neighboring cells that whether to what cellular identity they have to take upon, whether it has to make up the liver, it has to make up the heart, or etc. So this is the diagrammatic representation of the paracrine signaling, in which the cell is sending signal to the neighboring cell, or we can say the signaling cell is sending the signal to a targeted cell or a receiving cell. Next type of signaling which we do have is the endocrine signaling. So the endocrine signaling, for before going into that slide, you must know about the endocrine glands or know about the hormones. So what are the hormones? These are the chemical messengers. These are released by the endocrine glands. What are endocrine glands? These are the masses of the tissues which are ductless in nature. Ductless means they do not contain any ducts, or they do not contain any vessels from which the hormones can move. So the secretion of the endocrine glands are known as hormones, and these hormones do not travel through any ducts or any vessels. They directly uh, expel their secretion, that are hormones, into the blood. So from the blood, a cell moves towards the a signal, or a hormone moves towards a target cell. So the signaling pathway in which the messenger or the hormones are secreted by endocrine cells, they are transported to the distant target cells, unlike the previous signaling. So in this case, the cell targets a distant cell, or a much cell that is at the much larger distance through the bloodstream. So this signaling is known as the endocrine signaling. So mainly it travels through the blood. For example, the release of the cortisol from the adrenal gland. What are adrenal glands? These are present on our kidneys. So they release the cortisol. So this cortisol, the release of this hormone is stimulated by a hormone that is known as ACTH. That stands for the adrenocorticotropic hormones. This hormone is released from the anterior pituitary gland. This anterior pituitary gland is the example of the endocrine gland, which is I explained in the beginning of this slide. As it is an endocrine gland, so it does not contain any duct. So it will cause its secretion directly into the blood. From this blood, it will be taken to the desired target cell. So it is an endocrine product which occurs via endocrine signaling. At last, we have is synaptic signaling. So, a synaptic signaling, it is also the example of paracrine signaling, in which the nerve cell transmit its signals. So, it is the process. This process is named after the synapse. So, what is a synapse? Synapse is the point, or it is a gap between the successive neurons. So, it is a junction between two nerve cells where the signal transmission occurs. That's why it is known as the synaptic signaling. So when a sending neuron, or when a sending neuron fires, so whenever it starts taking its electrical impulse, it moves more faster through the cells. Hence, the signal travels down the axon. What is axon? It is a long fiber-like extension. It is known as axon. So electrical impulses always move along the length of the axon, from the cell body to the length of the axon. Whenever it reaches the synapse, it will trigger the release of the ligand. That ligand are termed as the neurotransmitters. So neurotransmitters are stored in the synaptic vesicles of the uh, presynaptic neuron, and they cross the small gap between the nerve cells. Whenever the neurotransmitter arrive at the receiving cell, that is also termed as the postsynaptic cell, where they have to bind, what will they do? They cause the chemical change inside the cell. That chemical change is often the opening of the ion channel, or that may be changing the electrical potential across the membrane. So let's uh, discuss it with the help of the structure. So, what is the synapse? Synapse is the gap between two neurons. So, this gap is termed as the synapse. So, the neuron which will be present before the synapse is known as the presynaptic neuron, and the neuron which is present after this synapse, it is termed as a postsynaptic neuron. But in case of a cell, it is termed as a sending cell. Sending cell is the cell which is sending the signals, and it is the receiving cell or the target cell which is receiving these signals. So these signals here are in the form of the neurotransmitters. Neurotransmitters are the chemical messengers which are stored in the vesicles, in the synaptic vesicle. So how this signaling takes place? First of all, these vesicles will fuse to the the membrane of the presynaptic neuron. After fusing, after merging with this membrane, it will release its contents, or it will release its neurotransmitter into the synapse, that is the space inside between these two neurons. So on the surface of the postsynaptic neuron or the other neuron, here the receptors are present. What are receptors? We discussed it in our previous section. These are the protein molecule which receive the chemical signals, or which receive the ligand. So after receiving this ligand, the response will be generated. So what is this response? As we have discussed, so after the receiving of the neurotransmitter, there will be a chemical change inside the cell, inside the second cell on which the ligand is binding. So this change is opening of ion channels. The ion channels may be opened, or second is the changing in the electrical potential across the cell membrane. So the membrane potential across the cell will also differ after the ligand molecule will bind to its receptors. So with that, the cell signaling is completed. So what we have studied in the cell signaling, there are different type of cell signaling terms. That first one is autocrine. What is autocrine? Autocrine signaling is the one in which the cell signal to itself. So we can also say that the autocrine signaling is the one in which the sending cell and the target cell, they both are the same. The same cell is sending signals and the same cell is receiving its signals. Second, we had the paracrine signaling. What is paracrine signaling? It is the signaling which travels a short up to a short distance. Example of this signaling is like the signaling between the neighboring cells which are adjacent or which are very close to one another. So in this case, one cell is sending the signal and the another cell is receiving the signal at a very shorter distance. Third type of signaling we had was the endocrine signaling. So endocrine signaling is mainly taking place at a larger, relatively very larger distances. So this signaling, in this the ligand molecule or the signal molecule travels through the blood. Why? Because endocrine glands directly expel their secretions, that are known as hormones, into the blood. So from the blood, these hormones travel into the desired places where they have to move on. So last signaling, we had the synaptic signaling. So what is the synaptic signaling? It is also the form of the paracrine signaling. But in that case, it especially involves the neuron. So in that case, the one neuron was the presynaptic neuron, other was postsynaptic neuron. So there was a gap between the neurons known as synapse. So these neurotransmitter, they act as the ligand here. They move from the presynaptic neuron to the receptors which are present on the surface of the another postsynaptic neuron. After the binding, there are very chemical changes which takes place. With that, signaling is completed. Now it's a turn for the questions. So question can arise from the this slide, such as what do you mean by cell division? Okay. So what is the cell division? A cell division is the process in which a parent cell is divided into the daughter cells. In its types, so what are its types? It has two main types. First is the mitosis and second is the meiosis. So in mitosis, only two daughter cell are produced from a single parent cell. Unlike in meiosis, there is the production of four daughter cells from a single parent cell. Next we have is differentiate between the reproductive cell and the somatic cell division. So for that, firstly we need to understand about the somatic cell and the reproductive cell. So what is a somatic cell? A somatic cell is the cell which is any cell apart from the reproductive cell. So a reproductive cell is the one which is also known as the sex cell. These are the spermatozoans or the oocytes. Okay. So this is the reproductive signaling involves the meiosis mainly, and the somatic cell division involves the mitosis mainly. So mitosis is a single step process and the meiosis is a two-step process. That's why in the meiosis four daughter cells are formed. Next question we have is define the homologous chromosome. So what do you mean by homo? Homo means same. So homologous chromosomes are the chromosomes which share the similar genes in the similar order. Next we have is the different phases of the cell cycle. So cell cycle contains two phases. First one is interphase and second phase we have is the mitotic phase. The interphase is the one in which no division of the cell takes place, and the mitotic phase is the one in which there is the division of the cellular contents takes place. So another we have is differentiate between the mitosis and meiosis, which I explained earlier. It is a one step process. Mitosis and meiosis is a double step process. Next we have is define cytokinesis. So cytokinesis is defined as a process in which there is the division of the contents of the cytoplasm from the one cell into the two different cells. So there is equal division of the cellular content, that are cell organelles or the cytoplasm of the cell. Next is explain about the cell cycle checkpoints. So what are cell cycle checkpoints? These are the regulators of the cell which regulate the normal activities or normal requirement of the cell, and they make the decision whether the cell has to divide or not. So there are different type of cell checkpoint: G1 cell checkpoint, G2 checkpoint, and the mitotic spindle checkpoints. So they occur at the different stages of a cell cycle. So next we have defined cell junction. So what are cell junction? Cell junction are the point where the two cells are held together by one another. So there are different type of the cell junction. They may be tight junction which are tightly held by one another. There may be gap junction in which small gaps are present, through which the movement of ions can take place, movement of or transport of any another substances takes place. Then we have the adherent junctions, we have desmosomes, we have hemidesmosomes, etc. Next we have is define cell communication. So cell communication is the process by which the cells communicate with one another, or it is the process by which a cell sends the signal to the another cell, or it sends the signal to its neighboring cells. So cell communication it has two types. It may be intercellular or it may be intracellular. The intracellular communication it involves the signaling between the cells, and intercellular signaling involves the signaling between the neighboring cells. So last we have is explain the different form of the signaling. So the different form of the cell signaling are autocrine signaling, paracrine signaling, endocrine signaling, and synaptic signaling. So with that, my presentation is over.